Let's come back to our case here and just recap some things that we've learned about Don. Don has had a CT scan and a PET scan of his head and his chest to determine if there's anything abnormal. And of course, they're very concerned that Don might have cancer given his years of smoking. And we'll continue to follow Don and Patricia through not only this discussion of DNA replication and the cell cycle, but also then how cigarette smoking might be interacting with his DNA. If Don does have cancer, then we need to start to understand a little bit more about what a cancer cell is and how a cancer cell could come about. So let's talk a little bit about where cells come from overall. We know that cancer cells, like any cell, is going to come from a pre-existing cell. So we also need to take a look at how a cell is formed, where it comes from. We know that all cells, whether they are healthy or whether they are cancerous, are going to arise from the process of cell division. And we're going to look at that part of the life cycle in just a minute. But what we want to try to understand as well is what is the difference between normal, healthy cell division. So, for example, right now you are making new skin cells. Some of the skin cells in your body are dividing and you're making new skin. You shed skin cells all the time and you're making new ones. You're also making new cells, let's say, in the lining of your digestive system, so lining your stomach and your small intestine. And that is healthy, normal cell division. However, we know that if things go bad with cell division, then we can have tumors form as a part of cancer. And that's what we need to explore, of some of the differences between those, and also how DNA is going to play a role in that. Let's take a look at a picture called our cell cycle. Our cell cycle is a sequence or series of events that take place from the time that a cell is formed until the time that that cell will divide. We're going to spend a lot of time on this figure a little bit later in our discussion, but right now I just wanted us to see the big picture. So here we can see that we've got two cells that have formed as a result of cell division, and if we picked this one cell and were to follow it through its life, eventually it would get ready to divide as well. So the events that you see in the cell cycle here represent what the cell will be doing. Now, when a cell gets ready to divide, there's a sequence of events that have to take place, and one of the most important happens right here in a phase called interphase, in S phase of interphase, and that is the DNA has to copy itself. If these two cells were made from one cell, it means that these two cells have identical DNA, and that means that there was an original cell that had DNA. The DNA copied exactly, then there were two copies that were separated, and each cell got a copy of that DNA. So before we jump into the whole big cell cycle and all of these events, I do want to talk about how it is that DNA goes through this process of replication. How can a cell take your DNA and make an entire copy of it? That's what's happening as you make new skin. That's what happens as you're making a new lining to your digestive system. So let's start big picture, and then we'll take a look at what happens when we get into the details of DNA replication. So that's what I wanted to address next. The model that we use for understanding DNA replication, that we will see in a lot more complexity on the following slides, is this. It's what we call our semi-conservative model of DNA replication. So if we look at that word semi-conservative, what that tells us is that we are going to start with our original DNA. So this would be in the one cell that's going to contain it. Let's call this the old DNA, because that's what some of the figures will term it. So if we start with our old DNA from our original cell, what we're going to do is actually use this old DNA as a template. And then from that template, we are going to construct new DNA. And so the new DNA that you can see here is in this blue color. This is new DNA. What we're left with then at the end of the process of DNA replication are two molecules of DNA. Here's one molecule of DNA. Here's another molecule of DNA. And we remember, I hope, that each molecule has two strands because it's a double helix. And now we can see that one of those strands is one old strand, original strand, and one is the newly synthesized strand in blue. Here is our second DNA molecule, and we can see the same thing. There are two strands, and one is the old strand, and one is the newly synthesized strand. So back to what this word semi-conservative means, it means that when a new molecule of DNA forms, it is half old and half new. It has conserved some of the original parent. Not all of it, but some of it. And that's why it's semi-conservative, one old and one new. So we're going to see this in a lot more detail. I just wanted you to be aware of this process as we move forward. So let's look at a little bit more detail with our picture of DNA and its division. So let's look at another picture that gives us a little more detail about how DNA can replicate. This is a picture that you have in your course manual, and if you have some different colors around, it might be a good idea to try to color some different areas of this so that we can show here our DNA replication with our old strand and with our new strand. So pick a color. I'm going to pick red, and we know that this is going to be our original DNA strand. So this was from our original parent, or that's the old. And we can see that our nitrogenous bases are held together here by hydrogen bonds in the middle. And then we notice that our parent, our old, is going to split. So if we were to color our old in red, here we can see that's occurring. And then we also know that we're going to have some newly synthesized DNA. So I'm going to put that in blue. This is the new DNA that's being synthesized. And what you'll notice is that it's using our original parent template as a code to follow. How does it know what to make? Well, we know that it's going to be using our parent, the old, as a template, and this side as a template, the parent, that's old. And then it's going to be constructing new DNA. So this is the newly constructed. So if we were to look at our final picture then, over here we have our two molecules of DNA. And if we were to color those, we know that we've got our newly constructed here in blue, and our older DNA is going to be in red, our parent. So we've essentially just made our previous picture, but I think it's helpful to see some of the intermediate steps as we go. I know this slide has a lot of detail on it, but I want to take us through the more detailed steps of DNA replication. What you'll notice first is our chromosome. So hopefully when we look at this picture we can see something that's familiar. And of course we notice our DNA double helix. And as we look at more and more detail, we can see our ribbon, which represents our sugar phosphate backbone. And then we can also see our nitrogenous bases, which are really colorful here. Any red is adenine, any orange is thymine, green is cytosine, and our blue is guanine. So let's check to make sure they got our picture right. We know that our complementary base pairs always should show a C going with a G. That means our green should always be with our blue, and that's what we see, for example, right here. And then we know that A and T are complementary base pairs, and we should always see them going together as well. This is important to remember because DNA replication is going to rely on this complementary base pairing, so we want to keep track of our individual nucleotides. All right, let's go through our steps. They are outlined in your course manual, and one of the first things that we have to do if we are going to use our parent or our old DNA as a template is we actually have to pull it apart. And so our very first step, step A, is that our DNA will have to unwind and we also have to break the hydrogen bonds holding our nitrogenous bases together. So we have to break the hydrogen bonds holding our nitrogenous bases together. And so this step A right here is being shown right here. You can see that that's where we're starting to pull apart our nucleotides, specifically at the nitrogenous bases. This area where they pull apart has a name. It can be called two things. Oftentimes we refer to it as a replication fork, Some books will call it the origin of replication. So you may see it in both ways as you watch different videos or look at different pictures. There's an enzyme that's involved in this process, and the enzyme that's involved in the unwinding and breaking of these hydrogen bonds is the enzyme called helicase. So you can think that if our enzyme helicase is breaking apart our helix, that might be an easy way to remember that. So that's our first step in the process of DNA replication. Our second step then is to start to use our old parent as a template and add new nucleotides. So we have to have a pool of our nucleotides. You can see here is a pool of free nucleotides. So here in green, this is a cytosine nucleotide, and in orange, a thymine. And this is just representing that your cell has to have them. And you can see more of these free nucleotides over here getting ready to be made into newly synthesized DNA. Well, where do these come from? Well, they come from a couple of places. Primarily, they come from our food. So, wait a second, we eat DNA from other organisms? Yes. When you eat an egg, you're eating cells from a chicken, and those cells have a nucleus, and that nucleus contains nucleotides. So when you eat them, you actually break them apart, and you will then incorporate them into your own DNA. If you have a piece of toast, then you're eating plant cells, maybe wheat. You're eating cells that have a nucleus, and those nuclei contain DNA, and you eat them. So these free nucleotides were at one point in another organism. So maybe this one was from the chicken that you ate yesterday, and this one was from the toast, and this might be from an apple that you had. So keep that in mind, that when you're constructing your DNA, these free nucleotides are things that in general we get from our food. There's also some evidence that cells can synthesize nucleotides as well, so they can actually make them. Both are explanations for where our DNA nucleotides come from. So how are these free nucleotides added to make newly constructed DNA? That leads us to our second step. We'll call it step B. And this is where we will have new bases added to the parental strand. So this is where we're using that parental strand as a template. And there's an enzyme that's responsible for doing that. The name of this enzyme is DNA polymerase. So remember, when we think about enzymes, enzymes frequently end in ase. And this is an enzyme, DNA polymerase, is simply making more DNA. The enzyme is shown here in orange, and you'll notice that it's a pretty big enzyme. It actually is going to hug the parent strand, and sort of inside of this enzyme, where you have an active site, the parent strand will fit. And then we're also providing a location to facilitate the chemical reaction of adding in new DNA nucleotides. But DNA polymerase has to follow those complementary base pairing rules. So let's take a close look to make sure DNA polymerase is doing its job. We notice that our parent is here on the top, and that our strand right here is our newly constructed. And so wherever DNA polymerase is going to read a blue right here, that's guanine, DNA polymerase will put in a C. And then our next nucleotide on our parent here in green is cytosine, so we should expect a blue guanine to be coming in, and it is. And then the next one is a red adenine, and we can see here that we've got an orange thymine coming in. DNA polymerase then is going to move along. You could imagine it like a little car wobbling along, and as it runs along our parental strand, these free nucleotides are going to be added in, and we create this newly synthesized strand. When DNA polymerase is wobbling along and making its way toward our replication fork, we call that our leading strand. The leading strand, as you can see, is going to be making new DNA continuously. When that DNA polymerase reaches the replication fork, then this area is just going to unwind with more of the helicase enzyme, and the DNA polymerase will keep going. So that's what we mean by the leading strand. It's the strand that's made continuously. Now, when you look at the other strand, the same type of thing is happening. It's just made a little bit differently, and I don't want us to get hung up on too much of the detail here. Because DNA polymerase has to go in a certain direction, and because our two strands of DNA have different directions to them, we know that DNA polymerase over here on this side has to move away from the replication fork, and it's going to be moving this way along the parent. Well, you can imagine that as it moves away, then we're going to have an area over here where we didn't have any of our DNA newly synthesized. So this DNA polymerase is going to have to keep jumping back to the replication fork and synthesizing a new piece, and then jumping back and synthesizing a new piece. What that means is that over here on this side, our newly synthesized DNA is what we call the lagging strand. And the lagging strand is going to be making new DNA. We could follow all of our base pairing rules. We could see A's going with T's and G's going with C's. But our lagging strand is going to be made not continuously, but instead made in pieces. Now, eventually these pieces will all get glued together. There's actually another enzyme that does that. The name of that enzyme is called DNA ligase. And this DNA ligase is going to join together all of these pieces. Incidentally, those pieces that we have of DNA that are going to form on the lagging strand are named for the person who discovered them, and they are called Okazaki fragments. So these Okazaki fragments only form on the lagging strand, as part of the lagging strand, and then DNA ligase is going to come along and put those Okazaki fragments together. The end result is the same. When we'll look at our finalized picture of our DNA molecules, we can't really see which one was the lagging and which one was the leading. Our goal is to just have two identical copies as we go. So that's something that I want to make sure we understand about these two. So our final picture and our final step here is that we will end up with two DNA molecules being formed. So let's look at a short animation that helps us see what that looks like. So if we started here with our original parent DNA, and then from that we had our enzyme, and that enzyme is helicase. It's going to come along and it's going to break apart our DNA strands. We broke hydrogen bonds, and this is still our parent DNA. And then, while it's not shown here, we're going to have a second enzyme come in, our DNA polymerase, and it's going to be adding in complementary nucleotides. And we'll do it one direction on one side and one direction on the other. So the end result is that we have two identical copies of DNA. Here's one molecule of DNA, and here is another molecule of DNA. And then if we got even more detailed, we would remember that because of semi-conservative replication, this right here, this half was our old parent. This over here was our old parent. And then we also have the newly synthesized, and that's going to be this right here. That's our new. And this is our newly synthesized. So we're able to, I hope, see back to some of the first pictures that we looked at, how we can use semi-conservative replication and end up with two identical copies of our DNA going through the detailed process that we've just looked at. One last thing I wanted to leave us with as we talk about DNA replication, and that is it always perfect? The question is do we always copy or replicate our DNA right? Are there any mistakes ever made? Well, the good news about DNA polymerase is that DNA polymerase has in it a proofreading mechanism. So it looks very carefully at the newly synthesized DNA to make sure that it's the complementary match to the original parent strand. So this is just one example where we can see here in our top frame that there's been a mistake. You'll notice our new daughter strands in green here are being synthesized. But right here, there was a G in our old parent, but the DNA polymerase put in a T. and this will get repaired. So if there's a mismatch, it can get repaired, and the appropriate one will be put in, and there'll be a C with a G instead. So DNA polymerase does have a lot of the proofreading. With that said, occasionally there are different types of mutations that can occur, and a mutation is a permanent change in the DNA. Later this semester, we'll talk about different types of mutations, and there are many different types, whether we're talking about just one base pair being mismatched, or maybe the mismatch isn't there. Maybe where there was an AT pair, it's now been replaced with a GC pair. Other types of mutations can involve large segments of DNA that have either been removed or duplicated or moved. So we'll address all of those a little bit later, but I did want you to be familiar with this term mutation here at this point because it will play a role in some of our other case studies. One last item related to the topic of DNA replication is taking a look at telomeres. This is something that's relatively new in science as we've learned more and more about chromosomes. As we take a look at a chromosome, we'll notice that at the end of a chromosome, we have this structure called a telomere. And a telomere is a repetitive sequence that doesn't actually code for anything. So this isn't actually going to be part of the genetic information that we require as a recipe for how to make a product inside of the cell. And the reason why I'm talking about them here is that as a cell divides, meaning every time DNA replication occurs, something interesting happens to the telomeres in that they get shorter. So when a cell goes through DNA replication and the cell divides, this telomere gets a little shorter. So that telomere is going to get shorter. And then if that cell goes through division again, then the telomere gets a little shorter. And we can see that over time, eventually, these telomeres get shorter and shorter and shorter. And we understand that this is likely related to aging. When we think about how old a cell might be, how old an organism might be, one of the things we can do is actually look at their telomeres. So if we took a sample of your cells from when you were 8 years old and looked at your telomeres, they would be much longer than if we took a sample of your cells when you're 80 years old and we looked at your telomeres. The purpose of these telomeres, we think, is to provide some protection for this area of our chromosome right here. We know that if there are telomeres there, then they are offering some type of protection so that some of the nucleotides that make up this DNA are not going to get sheared off or broken off. But as our telomeres age, it's going to put this area of our chromosome potentially at risk, and we oftentimes will see cells die once their telomeres get too short. So by understanding more about telomeres and what causes them to get short over time, we can actually learn not only a lot about the aging process, but also about some things related to cancer. And that's where we need to look next. So how could this be related to cancer? Interestingly enough, there's an enzyme whose job it is to maintain the length of the telomeres. So there's an enzyme, and it is called telomerase. There's that ace again on the end of a word, indicating that it's an enzyme. And this telomerase's job is to maintain the length of telomeres. Meaning that if you had telomerase active in a cell and the cell went on to divide, the telomere would not get any shorter. So it would essentially make that cell immortal, like the cell could keep going on and on and on and dividing if telomerase was active. So if we were to look at what cells actually have telomerase in them, it's actually an interesting group. First of all, we know that telomerase can be active in some fetal cells. So we realize that early on in an embryological development as well as into the fetus, that there are some cells and tissues that have telomerase turned on so that there can be rapid cell division as organisms grow. That's just a normal, natural part of the development in organisms. Another place where today, as an adult, we could look and find some telomerase still active in you would be in what we call adult stem cells. We haven't talked very much about stem cells yet. Oftentimes in the news we hear about embryonic stem cells, and these are not the same thing as we're talking about with adult stem cells. Adult stem cells are cells in your body that retain the ability to continue to divide over time to provide you with new cells. So let me give you a couple of examples. You have some adult stem cells in your skin. We know that you continue to make new skin every day. Like a conveyor belt, that skin rises up to the surface and it's going to slough off, and you continue to make new skin cells. So you have to have a population of cells that can continue to divide, and that will happen for the whole of your life. We know that another place for adult stem cells is in bone marrow. Right now you are making new red blood cells, and so we continue to make those throughout our entire life because a red blood cell only lasts about 4 months or so. So you have some adult stem cells there, and if we look, we actually find that the telomerase is still active in those cells. However, if we were to look at most of your body cells, we wouldn't see telomerase. The downside of telomerase is we know that it's often activated and active in cancer cells. So if we had a cell that was cancerous, bad cell, and it was undergoing cell division at a rapid rate, if it did not have telomerase there, then that cell would have an end date. We would find that that cancerous cell would divide and divide and divide, and then it would stop because the telomere would get shorter and shorter and shorter, and eventually that cell would die. However, if we look at telomerase, what we're seeing is that our enzyme is active, and that means every time that cancer cell divides, telomerase is going to keep the length of the telomere as long as ever. So this is really bad news, right? You have this cell that's cancerous, but now you also have this enzyme that is keeping that cell young. You have a cell that's behaving badly that is now going to essentially be immortal. It's going to divide and divide and divide over time as the telomere's length is maintained. So I wanted to mention some things about telomeres and telomerase here as we talk about DNA and cell division, because it's something that we hear in the news related to cancer and to aging, and even recently space exploration, as we've compared telomere length in astronauts versus those people who have not traveled to space. So take a look on Google and see what you find. Just to recap, as we look ahead, our next topic is going to be coming back to our cell cycle, and again looking at how a cell goes through these sequence of events. And hopefully we realize that as we've looked at one of these sequence of events, DNA synthesis, we know a lot more about how we can start with one copy of DNA and how a cell could duplicate that and now have two identical copies.